Everyone, my name is Cleidy Osorio. I am from Ribeirão Preto Medical School, University of São Paulo. I'm going to talk about my doctoral work titled Structural Characterization of the Central Region of FAZ10 Protein in Trypanosoma brucei. I am a member of this Laboratorio de Biología Celular y Molecular de Trypanosoma chidius, now USP, the principal investigator, Dr. Munira Baki. I would like to make a brief introduction about the disease, human African trypanosomiasis, or sleeping sickness. Sleeping sickness, this is a disease that disrupts the sleeping partners that we can see here. The diagnosing, we can see here, the diagnosing of this disease, including serological tests or lumbar punctures. About the treatment, this disease is determined including some drugs with intramuscular and intravenous injections, and some drugs with side effects such as melarsoprol, arsenic derivated drug. This disease is the symptoms can include fever, muscle pain, coma, even death. This disease is transmitted as caused the agent of the disease is a parasite Trypanosoma brucei with a unicellular. This disease is localized in the sub-Saharan Africa. And why? It's because the presence of the vector or ZZ fly that we can see here. We can see the complex life cycle of this disease, including the vector, the ZZ midgut, and the human host, mammalian bloodstream. We can see here that the parasite must change to adapt in different environments. Due to this characteristic and the impact in the human health, Trypanosoma brucei can be considered as a key model for biological research. This parasite is composed by two principal components, the cell body and the flagellum. Between these two components, there is a zone called flagellum attachment zone, composed by a lot of protein interacted between them to ensure the connection between the flagellum and the cell body. This flagellum attachment zone, or FAZ, can be divided into three domains: flagellum domain, intracellular domain, and cell body domain. This FAZ is very important for morphology, motility, and the survival of the parasite. One of the protein in the cell body domain is FAZ10 protein, a young protein with 2,000 kilodaltons, localized between the flagellum and cell body, and we can see here in this microscopy image, immunofluorescence. Firstly, this protein has been characterized for the first time in my lab. For the better understand of this protein, we can divide this protein, FAZ10, in 5 regions. FAZ10 is very important for the properly cleavage forum formation in the parasite and the correct distribution of the organelles in the cytokinesis, for a correct cytokinesis. Using RNAi to silence this protein, we can see here the importance in this graph, importance for the cell growth in the parasite. In absence of this FAZ10, the cell growth is low. And we can see here in this microscope image the incorrect cell division generating different phenotypes, as we can see here. Since we have demonstrated FAZ10 is important for the correct cytokinesis, my work aims to know about the structure of this FAZ10, to deepen understanding about the information about the function of this protein with two approaches, in silico and experimental characterization. About the experimental in silico characterization, we have performed disordered prediction with UPRE tool of all the protein. We can recognize here five regions, and we can recognize that central region has low disorder, so we can understand that this region has a smaller structure. Focusing in the central region as an initial point for our analysis, we perform a coiled coil prediction of this central region of FAZ10, and we have found one domain, coiled coil domain, within this central region, with a likelihood of dimerization as a parallel coiled coil. Using the recent Nobel-prized AlphaFold tool, we have predicted the structure of this central region of FAZ10. We can see here two globular domains flanking a one coiled coil domain with a length of 44 nanometers. Using the structure, we have recognized the amino acids composing the coiled coil domain. By literature, we recognize the A and D position of the domain, in this case with lysines, and in the position E and G, lysines and glutamines. The function of this protein hydrophobic amino acids in this position in red is in the interaction between the two chains, and in the function of this other position, polar amino acid, is to support this interaction. With these promising in silico results, now we want to confirm these in silico findings with expression of the recombinant protein by heterologous system of the central region at its globular domain. We can see here in this SEC purification, site exclusion chromatography purification, after the affinity chromatography, two peaks. This peak is the corresponding to molecular aggregates that we are not considering, and this peak number one is the central region is corresponding to homogeneous sample for the next analysis. In the case of the globular domain, we can see here other volume, other molecular size, in this case, a smaller molecular size, just the globular domain, that we can confirm by SDC page. 11 kilodaltons approximately for the globular domain, and all the central region, 66 kilodaltons. But until this point, we don't know about the dimerization of the central region. That is the objective of my work. So we have performed a small analysis of the recombinant protein of central region, including all the central region and its coiled coil domain and its globular domain. And we can see here in black the central region results with molecular aggregates in the void column. We are not considering this. And this peak with 144 kilodaltons that is corresponding to dimeric population. In the case of the coiled coil domain in blue is corresponding to 65 kilodaltons corresponding to dimeric population. In contrast to the globular domain with 12 kilodaltons that is corresponding to monomeric population. To know if this both result has consistency, we have conducted analysis by circular dichroism of this recombinant protein, central region and its globular domain. We can see here in both experimental and theoretical results the same minimal value in approximately 208 in the position 208 nanometers for both results in the case of globular domain. And in the case of the central region is the same, with a minimal value of 222 nanometers in both experimental and theoretical or in silico results. And it's very interesting to see that this minimal value corresponding to, in the case of globular domain, by literature, globular proteins, and in the case of the central region, by literature, proteins with high alpha helices in the structure. All of these find all of these results can lead this conclusion. In silico analysis, identified the central region as an initial point for our analysis. This central region has coiled coil domain flanked by globular domains, and the in silico experimental results talk the same thing. And talking about that central region of FAZ10 has a dimeric conformation that we can conclude that all the FAZ10 protein may also be a dimer. And our next step is to resolve this structure of the central region with techniques like X-ray crystallography. And the importance to produce this recombinant protein is to identify molecular partners of the FAZ10, to know more about the function of FAZ10 as organizer within FAZ and the relevance of this protein in the parasite. Thank you very much to my lab, da Facultad de Medicina Ribeirão Preto, na USP, and the Instituto de Física São Carlos in the laboratory of Richard Garratt, that he had the lab in São Carlos, help us with the biophysical analysis, the support, financial support of the agencies. And thank you very much to everyone for your attention. I'm happy if your questions, I am open for your questions, if you may have. Thank you.
We have any question?
Maybe you can work on the theory or more on the experimental, but do you know what theory the was used for the
ACD.
But at the end, comparing the ACD theory and experimental, they match.
ACD is in the acrylamide gel. That is I didn't understand about the ACDS page. No.
ECD, circular diagram.
Circular diagram. Sorry.
The simulation that you compare to the experiments.
Yes.
The LASCS. Do you know which level of simulations were done or which level of theory?
That our objective is to know if both experimental analysis are talking at the same thing. And the circular diagrams is to for the structure, circular structure, and the experimental recombinant proteins. We obtained the same minimal value.
Software or algorithm was used to simulate this?
Really, this is a collaboration with this institute, the Física São Carlos, and they know more about the algorithms. I don't know the information now about the algorithms using to perform the circular diagrams.
I think they were very happy with this kind of agreement.
Yes, it's very difficult, but we have obtained this, and we want to repeat this again, just to have best results to confirm our first finding, to confirm this minimum value for both approaches. Thank you.
Okay, thank you.